Hello, hello. Welcome back, everyone. My name is Kevin Curran, and I've put together a three-part video series on gene editing delivery. It appears we've turned the corner. We have gene editing tools. The question is, how do we deliver them into the right tissue of the human body so that we can gain access to all these different diseases, right? Diseases tend to express, become a problem in one particular tissue type. How do we reach these cells to make the necessary edits to help people out? So in the first two videos, in the first video I talked about the use of naked delivery or electroporation. We saw CRISPR and Vertex. These companies are going after sickle cell disease. They have the drug approved now in Europe and the US. They are electroporating guide RNA and Cas9 into bone marrow stem cells outside of the human body. That's, strategically, that's an easier target because you have the cells, isolated in a dish. I also talked about the use of viral vectors, AAV and lentiviral vectors to deliver gene editing tools. I'll touch on that in a second. Then in the second video, I touched about lipid nanoparticles. LNPs are great for in vivo delivery to liver cells. The question is, what other cell types can we hit aside from liver cells? So one idea is to use VLPs, virus-like particles, to try to go after some other cell types. And really, VLPs are sort of a blend. They've got some lipid nanoparticle properties and some properties of the viral vectors. So let's get into it here. Let's see how quickly I can move through this topic. So, Intellia, they are the most advanced company in terms of in vivo delivery. They are now in a phase 3 trial for ATTR. It's a liver disease, amyloidosis, and they are delivering guide RNA and RNA for Cas9 into liver cells with a lipid nanoparticle, and that's being injected into the blood. The blood's going to pile up on the liver cells, hepatocytes, and the LNP will move across the membrane, and that seems to be working really well. Liver cells, they like to take up these LNPs. So what's the ideal delivery system? It's got to be highly cell-specific. For gene editing, you just want a tight time frame, like a transient time frame, where the Cas9, the nuclease, is actually chopping the double-stranded DNA, right? That seems to be a goal. You don't want this long time frame because you might end up with chaotic, extraneous cuts. We want to be accurate and precise, make just the volume of cuts that is necessary to change the programming of that cell. And of course, we're talking about in vivo delivery. Ultimately, gene editing, it's got to go in vivo if we want the costs to come down, if we want the patient experience to be better, and if we want to be able to distribute into low and middle income nations where they don't have these highly advanced medical centers that can do all of these ex vivo steps. We want patients to come in to maybe a local rural clinic, get a single injection, and their tissue is reprogrammed, hopefully forever, right? That's the dream. They go about their lives. That's the dream. So how do we get there? Yep, we need to move beyond blood cells, immune cells, and liver cells. And we want to think about how to target other cell types, internal tissues. So, I think the way to orient with VLPs is to think about how they're different than traditional viral vectors that are being used for gene therapies. Lentivirus is great. It's been excellent for CAR T therapies for liquid cancers. It can hold up to 10 kb in size. That's sufficient for gene editing tools. And it's got pseudotyping. It's got these glycoproteins that gives a good amount of cell specificity, but it integrates into genomes, right? It integrates into chromosomes, and that's not ideal with a nuclease. We don't want long-term expression of a protein, of a Cas9 protein that can chop DNA. Problem with AAVs, they only carry 4.7 kb in terms of genetic cargo, and most, when a guide RNA and a Cas9, that tends to be, more than 5 or 6 kb. Also, AAVs create that circular episome in a nucleus. That is very stable. It dilutes out after a lot of cell divisions, but still, that may be more expression of gene editing tools than we want. That might be too long of a time frame where you have active expression of gene editing tools in a cell. They might start causing off-target edits, right? So those are concerns, right? Long-term expression may not be ideal. So enter the fray with VLPs. So VLPs, they promise all the cell-specific binding of a lentiviral vector or, an AAV serotype, and the capacity of a virus to transduce the cell, to enter into the cell, into an endosome, and then release the cargo into a cytoplasm, which can then, and then cross into the nucleus. So you want all those good elements of a virus, but you don't want the integration risk. You don't want long-term expression. You definitely don't want to move into a chromosome with your Cas9 system. So it allows that transient kind of tight temporal control or time frame for gene editing to occur. Okay, so here's a few VLPs. You've got the envelope glycoprotein here on top of a lipid envelope, and you've got the retroviral particle. You can put in the RNA can be packaged alongside these structural proteins. You can also create a pre-complexed guide RNA with Cas9 protein and have that ready to go. So that'll be delivered into the cytoplasm. So again, elements of lipid nanoparticles and elements of viral vectors. We're going to think about this glycoprotein, right? Because that's where, we're going to want to engineer that to gain access to new cell types. So we call that pseudotyping, that glycoprotein. The lentiviral manufacturing experts have been doing that for decades. So we have there's a lot of expertise there. So what I'm going to show you at the end here is just like thinking about mutating different glycoproteins, altering these traditional glycoproteins, and then possibly coupling those with antibodies to try to get, cell-specific VLPs that can gain access to new cell types. And I should point out, this is happening in a lot of labs at the early stage, drug discovery and animal work. As far as I know, there's no company that's entered into a clinical trial yet with a VLP for gene editing. If I'm wrong, please mention it in the comments. Let me know, and I'll take a look at that. I'd love to see what they're doing. But as far as I know, we're in preclinical. I was reading David Liu's lab. A recent paper there talks about the manufacturing. It is similar to lentiviral manufacturing. VLPs are formed by spontaneous assembly and budding of retroviral polyproteins encapsulating the gene editing cargo. So the ribonucleoprotein cargo is fused with the viral structural proteins. There is some deep history with VLPs. They've been explored as a platform technology for vaccines. So back in 1986, components of the Hep B virus were being manufactured into a VLP vaccine. So, you can see here, this is during the coronavirus pandemic. Here's the actual coronavirus. There's, the spike protein. So different groups were taking that spike protein and adding it onto the structure of a VLP. So similar idea. You're trying to deliver that chunk of the virus into the body, but without any integration risks or replication of the virus. It's just a false virus. It's a virus-like particle, but it's going to generate long-term immune memory to that spike protein. All right. So final slides, I just want to touch on, show you where kind of the front of this research is. A lot of groups, they're looking at this, the VSV-G glycoprotein. It's this vesicular stomatitis virus glycoprotein. You can see it's very proficient at allowing a virus to dock up and go through endosomal fusion. So, but it tends to be heavy towards certain cell types. So if you mutate that and then couple that glycoprotein with another antibody, then groups are trying to use that to gain access to different cell types. So here, this is a paper that came out this year from, Jennifer Doudna's group and these other folks. And they've got a VLP, and they're calling it an EDV, but as far as I can tell, it's a very similar idea. And they're using the VSV coupled with the antibody here, and they're going after CD19. That's a common antigen on B cells. And so they're just showing sort of proof of concept that they can deliver this virus-like particle into these cells, and they're bringing in a GFP protein to show they're moving into the right cells. So here they're thinking about T cell engineering. All right, so that's kind of where we're at with these VLPs. It's a lot of tinkering with like linkers on the surface attaching to the right, that the fragment of the antibody, the single chain fragment that has cell specificity, sort of coupling that with the glycoproteins to try to dock up and have high efficiency transduction of these harder to reach cells, move away from just liver cells or, in vivo and then just bone marrow and immune and blood cells ex vivo, right? So that's where we're heading, folks. That's going to be the next decade. So hit me up, like and subscribe, recommend other topics. As I have time, I'm going to keep making these videos. So as long as you folks enjoy them and they're helpful, I'll keep making them. I know a lot of the next generation of biologists and engineers seem to be watching these videos. So you're my target audience. That and just the general public who's excited about these topics and how they can help. All right, folks, I will see you next time.